So today we have the pleasure of welcoming. Do you want something or? Oh, okay. Of welcoming Christine Tardif, who is a newly appointed assistant professor at the Montreal Neurological Institute and in the Department of Biomedical or Biological and Biomedical Engineering. Or, you know, your biomedical engineering and then the programming. Professor Tardif and I go back certain ways. We were PhD students together, Bruce Pike. And after her PhD work, Professor Tardif was in Leipzig for a few years as a postdoc at the Max Planck Institute with a number of scientists there, mainly working at 7T on brain imaging. She recently returned to Montreal to work first at the Douglas Mental Health Institute with Malar Chakrabarty for
almost 2 years.
Yeah.
Almost 2 years.
And then, her recent appointment at the Neuro and in Biomedical Engineering at McGill brings her back to the McConnell Brain Imaging Center, where she's also a member of the core MR science group. And so today she's here to talk to us about her work in quantitative MRI of cortical myelin, promises of pitfalls.
Thank you. It's really a pleasure to be here and to see you again and see your work. So as you've said, my expertise is really in developing MRI acquisition and analysis techniques to look at the morphology and the microstructure of the brain. And today I'll really be focusing on my experience in imaging cortical myelin. So I realized you guys work on a lot of different parts of the body, so I thought I'd just, so that we're all on the same page to start, talk to you briefly about what myelin is. So myelin is basically an extension of an oligodendrocyte, which is a glial cell, okay? And it goes around and it wraps the axon, and it creates an insulating sheath. And so this is basically to speed up the conduction of action potentials along the axon, which basically connects distant areas of the brain. So you're basically just making, communication in your brain more efficient using this myelin. And it's also the reason why we call white matter white, okay? It's what gives it its white and actually kind of shiny appearance. And that's because of the high lipid content of the myelin sheath in comparison to other cellular membranes. So there's about 70% lipids and 30% proteins, whereas in typical or other cellular membranes, it's about the opposite, okay, in terms of ratios. And so when people think of myelin, they mainly think of the white matter, but there's actually quite a bit of myelin in the cortex as well. So about 10% of what you'd see in the white matter. And that's what I'll be talking to you about today. So in the field of brain imaging, there's actually a lot of people who focus on imaging myelin content, and I would say that one of the reasons for this is that it's one of the main sources of MR contrast, and it really does affect most of the contrast mechanisms that we use almost every day in MR imaging of the brain. So to start, I thought I'd talk to you briefly about why we'd want to use quantitative as opposed to qualitative imaging techniques to look at myelin in the cortex. And then I'll talk to you about a first experiment that I did imaging cortical pathology in multiple sclerosis, because I think it really illustrates some of the unique challenges that we have in imaging myelin in the cortex as opposed to imaging myelin in the white matter. So here we see a typical T1 weighted image of the brain, which I'm sure a lot of you have seen before, and it's really the workhorse of research and clinical imaging. A lot of toolboxes and pipelines have been developed to analyze these types of images, where typically what we do is we classify the voxels in the image to be part of different tissue compartments, so white matter, gray matter, CSF, and then we'll delineate the different structures and measure their volume or their shape. Okay, so this is typically what we do to look, for example, at the thickness of the cortex and how that could vary as a function of age or disease. And when we look at the cortex, it's kind of hard to visualize this information by looking at a 3D volume, so typically we also take it and project it onto a 3D mesh reconstruction of the cortex, as we see here. So this is actually the Desiccant Atlas. We're back. Okay. So basically, each label color here corresponds to a label, which is an area of the cortex, which is delineated by its shape. So the different gyri. But what about looking at the actual signal intensity? People tend to focus always on shape, on volume. But there's actually the signal intensity that you see in the gray matter actually contains information about the microstructure of the tissue. So that's what I was interested in looking at. So backing up a little bit, what is T1? Maybe all of you know this, but it's a longitudinal relaxation time. Okay, so the time that it takes for your longitudinal magnetization to return back to its equilibrium position. So, for example, as you know, in MR, we're looking at the spins, so nuclei with an odd number of protons or neutrons, which are spinning in your body, and they're all oriented in different directions. And as soon as you submit them to a main magnetic field, they all align with or against that main magnetic field, and you get a net magnetization, right? And so when we give energy to these spins, when we excite them, they deviate from their equilibrium position into the transverse plane. And as they recover back to their original longitudinal magnetization, the time constant T1 characterizes that recovery. Okay? And they're basically going back to that equilibrium position by interacting with their environment, which we refer to as the lattice. So what does this mean in the context of the brain? What are the sources of T1 contrast in the brain? Well, there's quite a few. The first one is, not so the first one, but one of the main ones is the macromolecular content of the tissue, so the proteins and lipids that are there. And in the case of myelin, as I said before, it's really enriched in lipids, so that will be one of the main sources of contrast, which really here contrast the white and gray matter of the brain. But there are other things that contribute. So proton density. So just how many protons are there that are contributing to the signal. So this means that, for example, you'll be very sensitive to edema. Axon caliber, this is something that people don't realize, but you're actually looking mainly at the protons that are on the outer sheath of the macromolecules are on the outer sheath of the myelin, so you're really sensitive to axon caliber. Iron also contributes, as well as dissolved oxygen in the tissue. So I guess the message here is that T1 is actually sensitive to the myelin content in your tissue, but it's definitely not a specific measure. And it gets worse. So when you're looking at a T1 weighted image, the signal intensity that you get is not only a function of T1, there are other tissue parameters that will contribute as well. So although to a lesser extent, such as T2 star and proton density. And then obviously your signal intensity is a function of the sequence acquisition parameters like the echo time, the repetition time, things like this. And in addition to all that, we also have methodological biases, which can cause some problems. The main one that we're concerned with in T1 weighted imaging is the non-uniformity of the B1 transmit field. So for example, down here, we see a B1 field at 1.5 Tesla and at 3 Tesla. And as you can see, the non-uniformity of this field just worsens as the field strength increases. And that will basically cause not only variation in the signal intensity in your image, but also in the contrast, okay, as a function of location. And so what happens is that here, for example, you have 3 different T1 weighted acquisition sequences, which are differentially affected by A1 non-uniformity. And that will then have an impact, for example, on the gray matter segmentation as a result. Okay? So this is an important confound because basically it's about the system. It's not about the tissue, and that's really what you're interested in. Okay? So as an alternative, what we do is that we measure T1 directly. To do that, you acquire several T1 weighted images where you vary the acquisition parameters, and then you basically fit that to your signal equation. And there you get a T1 map where you have a quantitative value at each voxel, and now you can directly compare this value between subjects, between time points, between systems, and things like this. So I won't get into detailed pulse sequence design today, but needless to say, there are many different ways of measuring the T1 relaxation time. The gold standard is the inversion recovery sequence, where you basically invert your signal, and then as it recovers, you measure it at a specific time, and you do this repeatedly for different inversion times, and then fit that to your exponential equation. It gives you very nice and accurate maps. The issue is that it's very inefficient. So you're looking at low resolutions, poor spatial coverage. There are different techniques that are much more efficient than this. When we were in the Bruce Pike's lab together, Eve actually tended to use the look-locker sequence, which, as you can see, is actually quite close to the gold standard inversion recovery. I tend to work with the variable flip angle sequence. And there is a good reason for this. Yves was doing T1 mapping in the context of his quantitative magnetization transfer experiments, which are much longer experiments, a much more detailed characterization of the MR parameters of the tissue. And he was doing this to look at MS pathology in the white matter. Okay, so he was looking at, more limited field of view, let's say, and coarser resolution. I was interested in looking at pathology in the cortex, and I didn't even know where it was. So I needed to go at higher resolution and look at the whole brain. So I cannot use that technique, and that's why I tended to go with variable flip angle. But the whole point is that this is the same subject, the exact same slice, the exact same scanning session. So these three maps should be exactly the same, but they're not. So there's really a trade-off in your acquisition techniques between the accuracy that you can achieve and the speed or resolution that you can achieve. So, as I mentioned, the driving force me for using VFA was to look at cortical pathology in the cortex. And we really made a tremendous amount of progress over the last almost 12 years now. So when I started working in MRI in 2006 in Bruce Pike's lab, we were acquiring 1 millimeter isotropic T1 weighted images at 3T. And that took roughly 10 minutes, 9-10 minutes. Okay? So now, in about the same amount of time, we're acquiring 0.7 millimeter isotropic T1 maps at 7T. And this is really due to a lot of improvements in the hardware and in the software, so increasing magnetic field strength, in the number of coils, channels that we use in our coils, better pulse sequence design, etc. So really, as a field, a lot of different changes have accumulated to this. So I'll kind of jump into our experiment on imaging cortical demyelination in multiple sclerosis, because it uses these high imaging, high resolution techniques, but it also highlights some of the other challenges that we have by looking at myelin in the cortex. So most of you may have heard previously of multiple sclerosis. A lot of people think it's a disease of the white matter, that you get these lesions that are typically inflammatory at first and then become chronic, and they're really obvious on MR images, such that MR is now a part of the clinical diagnosis pathway. Okay, but actually we've known now for over a century, really, almost from the beginning, that MS really affects the entire central nervous system, not only the white matter. The problem is that we only see it in the white matter when we scan a subject. Okay, and this is mainly due to the different pathophysiology of the gray matter lesions in comparison to the white matter lesions. So first of all, there's a much smaller inflammatory content in the gray matter lesions. You do get, really the hallmark of gray matter lesions is demyelination, but the effect size is much smaller because you have less myelin to start with. They're also remyelinate much more quickly, and so you have a smaller time span at beginning to detect them. And we also have the problems of just the geometry of the cortex. So here we have, the lesion types in MS of the cortex. Can be categorized into 3 different categories. You have type 1 cortical lesions that are really mixed white matter, gray matter, and those are easier to detect because of the white matter portion of it. Then we have the type 2 small intracortical lesions, which are really difficult to see and tend to be around a blood vessel. And we have the type 3 subpeel lesions, which are actually really thin. They only affect the superficial peel layers, but they tend to be cover a large area. Okay, and so in a lot of these situations, except for type 1, but in type 2 and type 3 lesions, we have problems with partial volume effects. So unless we get a high enough resolution, we won't be able to detect these lesions. So we decided to address this by doing a post-mortem imaging study of a multiple sclerosis brain. So we got this sample from the Douglas Brain Bank. They were fantastic to work with. They allowed us to scan the whole brain when they received it and to then kind of collaborate with them to do all of the histopathology afterwards. And so we basically decided to, scan this brain over the weekend on our 3T scanner, acquiring several contrasts. We achieved a really nice resolution. And to then kind of look at what the quantitative MRI signature of this pathology is and relate it back to the immunohistochemistry. Okay. So we have some, so this is basically a stain for myelin basic proteins, so we can see the myelinated fibers in the brain. So the myelin is basically these like brown fibers that you see, okay, and the blue is kind of the background, counter stain. So on the top row, we actually have a section of healthy gray matter from a control brain, okay, where you really see those fibers in the white matter go all the way up to the cortical surface, right? And you see maybe this dark band, which is the band of Voyager. That's a band within the cortex that has a denser myelination. Then on the second row, we have a slice from the normal appearing gray matter of our MS brain, where again, you see that nice myelination. On the third row, you have a subpial cortical lesion, which tend to also often occur in the fundus of the sulcus. So where the red line really delineates the boundary. So we really have, this column is a zoom in of what you see there. So you really see that there's absolutely no myelin left in the superficial cortical layers. So what we decided to do was to basically segment these high resolution images into basically myelin, no myelin, and then decimate them to get a map of myelin content. Okay. And to compare this, align it with our MR data, and then do a comparison. So here we have that same section where the black line delineates the lesion, and here are our different MR maps. So a map of the T1 relaxation time, of the relative proton density, of the T2 relaxation time, and magnetization transfer ratio. Doesn't matter if you don't know what all these means. Suffice to say that they're different MR contrasts that are all sensitive in different ways to the myelin content of the tissue. And I was actually really happy when I first saw these results. I thought you can really nicely delineate the lesion, especially in the T1 map. That's the one that had the highest correlation, actually, with the MBP stain. And so it kind of created some doubt. I was like, why do we have so much trouble seeing these lesions in vivo? Of course, there's the issue of the resolution, but still, if we fix that, shouldn't we be able to see them? So I started looking at the data in a little more detail and plotting the histograms that I saw. So basically, I had about 10 different cortical lesions, and I drew ORIs in those lesions and in the neighboring normal appearing gray matter. Okay? And so you see those histograms there in blue and red, and yes, they do overlap a bit. So this could, explain some difficulty in delineating or detecting these lesions. But then, if you actually look at the histogram of the T1 times over the whole cortex, you get that beautiful, smooth, black curve. No two peaks, so it's really difficult for an algorithm, for example, to segment lesions from normal appearing gray matter. So I started to wonder why that is. And if you go back to some of the classic myeloarchitectonic work done by the Vokes, this is in like 1910, and for the following 50 years, they drew basically the myelin of the cortex in different cortical regions. Okay, so these are representative of different regions of the brain, going from the white matter surface to the peel surface. And what you see is these radial myelinated fibers that go up from the white matter into the cortex, and then some tangential bands as well. And depending where you are in the cortex, there's a lot of variability there. So basically, this is kind of, drowning the signal that I had. How can I tell apart a lesion from normal appearing gray matter if there's the same amount of contrast just in normal, healthy cortex? So this was one of my concerns. And going a step further, if I actually took my data and plotted it onto the cortical surface, this is actually a T1 map, so you don't have any spatial non-uniformities, right, due to the B1 field, okay? Those have been removed. You actually see quite a lot of variation across the cortical surface, and this is actually normal for the most part. Some of the red areas may be lesions that tend to be in the sulcal fundi, but at the top, those areas in dark blue correspond to primary cortical areas of the brain. So primary motor cortex, primary visual cortex, and auditory cortex is kind of hidden here. But those areas have much higher myelin content, and so that's why you see them pop up here. So this actually kind of, so it has been validated using MR imaging of marmosets, followed by extensive histology all the way through their brain. This work was done by Nicholas Bach, who's now at McMaster University. So here we have a myelin stain, where again you see the nice band of Voyager in the cortex, denser myelination area MT, which is a visual area, very myelinated in humans, but even more so in non-human primates. And you can kind of see this, these features in the T1 map as well. And this is a T1 weighted image in this case, plotted on the cortical surface of this marmoset. And again, you see those primary areas. So B1 for the primary visual cortex, MT, A1, that's primary auditory cortex, somatosensory cortex, and the frontal eye field. So areas that we know have a denser myelination than the rest. So this really, in those years led to a whole wave of people who started to do cortical myelin mapping using different MR contrasts. So some of the ones that I showed you in the MS brain earlier. So I focused mainly looking at T1 maps. The group in London also looked at, well, R1, so 1 over T1 maps. There's the Human Connectome project led by Glasser and Van Essen, who looked at T1 weighted over T2 weighted contrast. Jean-Claude Haddad, who looked at T2 star maps. And I've also seen work looking at MT-SAT on the cortex, as well as more recently, diffusion imaging of the cortex that also shows this contrast. So it's really, I think, a field that has emerged over the last couple of years. And at that point, I was wanting to start to work at ultra-high field because I was so enthusiastic about the post-mortem images that I'd acquired. I thought they were so beautiful compared to the in vivo images that I wanted to work at ultra-high field to acquire higher resolution images of the cortex and take this one step further. So you're probably not all familiar with the Brodmann Atlas. No? Okay. So the Brodmann Atlas is really the most popular atlas that people use to know where you are in the brain. Okay. It's based on the cytoarchitecture of the brain. So basically, a scientist in 1909 who really went through histological sections and looked at what type of cells were there, how they were organized, and based on that, start to delineate boundaries between different cortical areas based on their structure, but also this was supposed to reflect their different functions. Okay, so it was divided into 54-ish regions, I believe, and this was then now put into common, space, into MRI space. So people use this, align their functional MRI data to this atlas to know where they are in the brain. Okay, so heavily used by the functional imaging community. A bit of neuroanatomical history. This was actually just supposed to be a first draft. Dr. Brodmann was actually a disciple of the Vokes. Okay, and the Vokes, they were more interested in the myelin. So they were the ones who were doing these drawings, okay? And they wanted Dr. Brodmann to basically do a first draft. It took him a year or two. It took them and the Vokes and their disciples about 50, 60 years to come up with a whole mapping of the brain into about 200 areas. And because that was done by different scientists who worked together, it was published in a lot of different articles, and it's only recently that Dr. Nieuwenhuis actually brought them all together into a common space. Into MNI space as well, and that was published in 2016. So I've been reading a lot about this, classic histological approach to mapping the brain, and I thought, well, if I can acquire high-resolution images of the cortex and start to see this contrast between cortical areas at different depths, maybe I can use a similar approach to start to map the brain of individual subjects and know where we are in individual subjects. So that was really the objective. So to be able to do this in vivo, so this is actually a map of 10 or 12 subjects averaged together. Okay, and you can see that nice contrast between cortical areas. But really what we wanted to do is to be able to do that in individual subjects, as we show here. Okay. So that falls into, this next step where really I developed some techniques here, actually mainly analysis techniques, to start to analyze that high resolution data. So I moved to Leipzig, and there they were already had been working at 7T for several years, and started working with the MP2H sequence that was actually developed by Marrakech. And it allows us to acquire T1 maps at 7T that have minimal B1 effects. And so here we have a 0.5 millimeter isotopic T1 map. You can start to see, let me move it up here, for example, the stria of Gennari, okay, in the whole brain in vivo T1 map of the subject. To give you an idea, we were first able to see the stria of Gennari in an MR image in 1992, so a long time ago. But this was done with a surface coil at 1.5T in a weighted image, not a quantitative image, you know, with nice thick slices at the perfect orientation to get the stria of Gennari. Now we're talking about a whole brain quantitative T1 map at 0.5 millimeter isotropic. So it's really completely different, a huge gain in information. So the next step is really to be able to build a coordinate system that fits within the cortex so that we can start to look at the different layers, the same way the books did. So I had the pleasure of working with a couple of really amazing scientists, including Dr. Pierre-Louis Bazin, who had basically decided to refine a lot of image processing tools to work on quantitative data sets as opposed to working on the traditional T1 weighted images. And so he basically worked a lot on the image segmentation to really start with accurate boundary surfaces, delineating the white matter from the gray matter and the gray matter from the CSF. And this next step was really the crucial point. Miriam Vaynert, a PhD student whom I mentored, she developed a new layering technique so that we can delineate these intercortical surfaces, okay? And it was based on an early observations by Dr. Bach, who observed that basically if you, that cortical segments will basically adjust their width based on the curvature, okay? So here we have the white matter surface, the peel surface, and if you just follow that yellow box, you'll notice that the width of it changes depending on the curvature of the cortex, okay? And that he observed this actually by cutting different sections and weighing them and seeing how many neurons and everything were in each. And so what Miriam decided to do was to basically implement this equivolume technique to define these intermediate cortical layers. So it's purely a geometric model. It does not rely on the image contrast at all. You just need to feed it in the boundary surfaces and it computes it from there. And she validated this technique using high-resolution postmortem images. So this is actually from the primary motor cortex, I believe. And again, you can see the nice two bands of Baillet. So that's a nice way to really know exactly where your cortical layers are, your true anatomical layers are. And here we have three different layering approaches. The most common ones used to date were the Laplace approach and the equidistant approach. And if you just follow that red line, it does not like hug the true cortical layers at all. You see here how it just doesn't follow it at all. The equidistant approach does a much better job, but it's really only the equivolume approach that nicely follows the bands of Baillet all the way through. And it does not rely on that intercortical image contrast at all. We just feed it the boundary layers. It's a geometrical approach. So we wanted at this point to see what impact that layering technique would have on our profile analysis. So by profile, I mean we're looking perpendicular to the layers, kind of like the way the Vokes did, you know, so the radial fibers coming up. We want to trace those profiles. So again, this time we're looking at a postmortem section of the primary visual cortex. So you see the stria of Gennari there, and we did the three layering approaches. And here we have in blue cortical profiles that go through the gyral crowns, and in yellow profiles that go through the sulcal fundi. Okay, so basically two extremes in terms of cortical curvature. Yeah? And the dip, the single dip that you see here, basically corresponds to the stria of Gennari. And so if you look at the Laplace approach, these dips do not overlap at all. So as you go through the cortex, you're not sampling the same layer. You're jumping from one layer to the next. Using the equidistant model, you bring them much closer together, and the equivolume, they're basically overlapping. So this means that if you choose a region of interest that has a lot of cortical curvature, which in the cortex you can't really get away from that, you're at least comparing apples with apples. You're staying in the same cortical layer all the way through your region of interest. So far we've kind of validated this using post-mortem MRI, beautiful resolution. This was like 75 micrometers isotropic, scanned at 7T for a very long time. Is it worth it in vivo? It is. So again, here we have a T1 map that's mapped onto the cortex, onto a cortical surface, extracted using the equidistant technique and the equivolume technique. I'm not even showing you the Laplace because it looked absolutely terrible. And as you can see, we have like these, so this is the surface. Here we blow it up so you can see the information more clearly. And as you can see, there's like these artifacts here, these black lines, which are really come from the top of the gyre. And that's because when you arrive there, you're basically jumping from one cortical area to the next. If you use the equivolume approach, these artifacts mainly disappear, mostly disappear. So actually, this was really of interest to the people who are doing cortical mapping, structural analysis, but it was also a huge win for the functional MRI community, because there's a lot of people who are trying to do high-resolution functional MRI at 7T, and what they wanted to do was differentiate the cortical layers because they wanted to show the difference between the input and the output layers. So this was actually a huge win for them as well. So the last step here is that what I showed you was for an individual subject. So a 3D intercortical grid that's defined for an individual subject. But we want to, of course, do some group analysis. We need to bring these subjects together into a common space. So typically, the way people do that is that they align cortical surfaces between subjects based on their geometry. Okay? And we do know that there's a strong relationship between the shape of the cortex and the cortical areas. So the visual, primary visual cortex is always in that calcarine fixture, right? But this relationship, even though it's strong, weakens as you move towards higher order cortical areas. And so even if you align brains based on their shape, some areas will be misaligned in terms of their function. And so what we developed is a new surface registration technique where instead of using only information about the structure of your brain, like the shape of your brain, we also added this T1 contrast on the surface, because that could help you basically really align the cortical areas one over the other. So this is basically the end result. This is an average of 10 subjects. It's a 3D T1 map at different cortical depths. So we're going from the peel surface here down towards the white matter surface, where you have a nice gradient in T1 times because there's higher myelin content towards the white matter surface. So they're all windowed the same on the top two rows. On the bottom two rows, each layer is windowed differently to kind of highlight the contrast of the different layers. So for example, if we're looking at differentiating primary motor from primary sensory cortex, we'll go towards more superficial layers because you can nicely see the contrast here. If you're interested in looking more at the primary visual cortex and deleting it from V2, then you might want to go down here. This is where your primary visual cortex is, and on the side here, that's V2. So really showing that you have additional, richer information by including this depth component. So this is just to say that I think that we went, we really went into the direction we wanted to go into, which was really to mimic some of the classic neuroanatomical work. Now we can look at profiles from different cortical areas and really see how they differ, which means that we can start to look at the relationship between structure and function in individual subjects instead of having to rely on that Brodmann atlas. Okay. So this is really work that I did mainly in the group of Bob Turner at the Max Planck Institute. But while I was there, I also worked quite closely with the neurology group. And they were interested in using these quantitative imaging techniques, but for a very different purpose. They were interested in mainly brain plasticity. Okay, it was a group that focused a lot on stroke and motor learning, and learning about brain plasticity mechanisms so that then you can enhance recovery after stroke. And by spending time with them and learning about brain plasticity, I learned a lot about myelin and its role in brain plasticity. So, the first two lines are what everyone who's writing a thesis in MS or on myelin writes in their thesis, right? What does myelin do? It helps you achieve and maintain rapid and energy-efficient conduction of action potentials. This is what I told you on the first slide. It also provides metabolic support to the axon. That's a little bit newer, but we've known it for several years. But by discussing with this group who's really interested in brain plasticity, I found out a lot of new things about myelin, which is really like new information. These last couple of studies are really groundbreaking work done in the field of like animal models and things of the sort. So we now know that, for example, myelin is responsible for the fine-tuning of the syncopated neural activity and modulates the magnitude of oscillations. So, for example, if you think of the way your brain functions, and it's not only an area alone, right? It's a lot of areas that are working together. And if you do EEG or MEG, you'll see that there's these nice oscillations at different frequencies in your brain. And it's the myelin that's responsible for fine-tuning the arrival of action potentials to really achieve that synchrony. Okay. It's also a very dynamic process. So yes, most of the you myelinate very fast during their first 2 years of life, such that by the age of 2 you get the contrast in the image that you see in an adult. But your brain continues to myelinate throughout your life, and you actually only achieve a peak in myelin content, I think, in your 30s or 40s. Really exciting new studies is that myelin is modulated by neuronal activity. So if you literally stimulate an area of the cortex, you will produce new myelin sheaths there. People have also shown that it's modulated by environmental experience. So if, for example, you isolate animals such that they have no contact with other animals, they will actually have hypomyelination of their cortex. And a very interesting new study that came out in 2014 that myelin is required for learning. So in this case, they were looking at animals and teaching them a complex motor skill. And if the animals were not capable of forming new myelin sheaths, they weren't able to master that skill as well nor as quickly. And so this, for me, made me realize that we wanted to use these quantitative imaging techniques to study brain plasticity, and that maybe myelin actually had a role to play there. In addition to that, as Yves said, after my experience in Leipzig, I came to Montreal and worked at the Douglas Mental Health Institute with Millar Chakravarti. And in my experience there, I realized that a lot of mental health disorders are characterized by subtle deficiencies in myelin. So we're not talking lesions anymore the way we see them in MS. We're talking about subtle hypomyelination of specific tracts in the brain, which lead to, significant differences in behavior and in cognition. So this is really, what I think drove the development of my own research program, which is to continue to look at the myelin content in the brain, but for a very different objective, to look at the relationship between myelin plasticity and complex behavior or cognition. And so, but my strength remains in the development of new techniques. And so one of the things I realized that remains a challenge is the lack of the specificity of our imaging techniques. And that's something that we really want to address in the lab. And so just to kind of illustrate this to you, I'll give you an example. So back to that group at the Max Planck, they were looking, one of their main interests was to look at short-term brain plasticity, okay? So in a very short time period. So they would do really beautiful imaging studies where of a subject where they scan them extensively, have them do some type of complex motor learning, these balancing systems, and then scan them immediately after, and then a week later and a month later type thing. And they sometimes saw differences mainly in the volume of the gray matter of different cortical areas, but they had trouble understanding what this meant. And there are beautiful review articles, including this one that was actually published by Robert Zatori, who's at the Montreal Neurological Institute. It's my favorite review article on the subject, that kind of explains, what are the different biological mechanisms that could lead to this change in gray matter volume that we see using MRI, and kind of, really highlights the need to improve the biological specificity of our techniques. So this brings me back to this slide that we looked at earlier. Even if I use quantitative T1 to look at the myelin of the cortex, in the maps I showed you earlier where we're contrasting different cortical areas, I was pretty confident in saying the main source of the contrast there is myelin. But if I see change in T1 in subjects over a period of time because they're learning a new skill, I'm not as confident about my interpretation because of this. And so this is where I actually teamed up with Dr. Claudine Gauthier, who was with me in Leipzig at the time, is now a professor at Concordia University here. She was interested in motor learning and has a lot of her expertise is in quantitative MRI of brain physiology. So what we did here is that we decided to use breathing challenges to simulate the type of things that we think would happen in the brain during motor, if you wanted to look at brain plasticity due to motor learning, over the lifespan, or due to differences in cardiovascular health. So the subjects were put into the scanner and scanned over a period of approximately an hour, during which they had 2 breathing challenges. The first one was a hypercapnic challenge, so you increase the amount of CO2 that they're breathing in. And this will increase the amount of cerebral blood flow and therefore cerebral blood volume in your brain. And our hypothesis is that this would actually have an impact on the cortical gray matter volume that we measure using these T1 images. Okay? The second breathing challenge was one of hyperoxia. It's less of a challenge. You're breathing basically a higher concentration of O2, and this would lead to a higher concentration of dissolved O2 in the tissue, which is a paramagnetic agent, and therefore it would decrease your T1. So they were scanned first breathing ambient air. They would start breathing in, for example, the higher concentration of CO2. We'd wait for the effect to stabilize, okay? And then we would acquire a second T1 map, back to ambient air, and then the hyperoxic challenge. So this is what the T1 maps looked like. This is for the ambient air, and there I'm just showing you the zoom in of that area for the different challenges. And they look almost exactly the same. That's the point. So you wouldn't expect there to be big difference. You know, people will look at these images and be like, there's no bias, there's no problem, moving on. But actually, once you analyze them in a systematic way, there are significant differences. So first, looking at the hyperoxic challenge, if you compared it to the first ambient air, there is a decrease in your T1 at 10 milliseconds, whereas if you looked at both ambient air challenges, there's no difference at all. And here each dot represents a different subject, and you can really see that it's just a shift downwards, a shift, a decrease in the T1 time for all of the subjects. This is averaged across the entire cerebral cortex, okay? But the effects are actually quite uniform and widespread. So here we're looking at the differences, across the whole cortex. We see them there, and it's actually very symmetrical as well. I have to admit, I wasn't very worried about these changes. We're talking a 10 millisecond difference. It's not huge, but we still need to take into account if you're looking at a short-term, effect due to a cortical cognitive task or due to motor exercise or something like this. If you take it into the context of normal aging, so there's not a lot of data on this, but if you take this data set that was published by Dr. Callahan, by making them breathe in O2, we basically make them look about 2 years younger. That's the effect in terms of the T1. It had a much more significant impact on cortical thickness and cortical volume. Okay? So here we were doing a hypercapnic experiment, again, increase in cerebral blood flow, which leads to an increase in cerebral blood volume, and a difference in the size of your gray matter compartment, basically. So we have basically a change, an increase in cortical thickness of 1.85%, an increase in gray matter volume of 3.3%. Okay? And this is actually really big, considering that the cerebral blood, like the vascular compartment of your cortex only represents 5 to 10%. So that's a huge change. The hypercapnic challenge that we gave the subjects actually caused a CBF increase of 30 to 38, CBF is cerebral blood flow, so it caused a flow increase of 30 to 38%. So this is actually equivalent to the type of increase that you get if you're doing a visual task. So if you're just looking at a checkerboard while you're in the scanner, these are the types of CBF increases that you'll get. If you're doing a cognitive task, it's more on the order of like 15 to 20%, okay? But just to tell you, this is like a normal physiological range. And if, again, you take this into the context of normal aging studies, okay, Salat published data where he looked at cortical thickness differences over the lifespan and CBF differences over lifespan in the same subjects. And if you put that together, it means that differences in cerebral blood flow and volume would basically account for 9 to 30% of the changes that we see in cortical thickness. And when people publish data where they say, oh, I have a decrease in cerebral blood volume, gray matter volume, they automatically interpret like neuronal loss. But it could be really, and given if you have a significant decrease in your vascular bed, that may lead to neuronal loss, but it impacts your interpretation of the data. And again, Alzheimer's disease has a decrease in cerebral blood flow of up to 40%. So if you see a decrease in your volume of 3%, it may simply be due to the flow, the volume, and not due to neuronal loss. Again here, just showing that these effects were very uniform across the brain, affecting both hemispheres quite symmetrically. Here looking at the cortical thickness differences, and here at the gray matter volume differences. So there are some small abnormal areas that showed up in the cortical thickness that you see in blue, especially in the calcarine surface or really in the midline. And we think that this is mainly due to the large blood vessels that you find there that may be swelling and therefore may be preventing the cortex to get thicker in a way. And so it may just be affecting more the surface area as opposed to the thickness, because we do see differences in gray matter volume in those areas. Okay? So this led to a review paper that Claudine and I and a couple of our colleagues at the Max Planck wrote together to really kind of highlight some of the advanced quantitative imaging tools that we could use to gain a better understanding of what's happening at the microstructural level, specifically in the context of, brain plasticity studies. But one of the main points that we try to address in this review paper is that there is no direct link between the MR parameters of our tissue and the microstructural features that we're actually interested in. Okay? And I think that the MR community kind of as a whole is deciding to address this issue by acquiring multi-modality data. Okay? So multiple contrasts. And that's a direction that my lab is going into, which means that if we're going to put a subject in the scanner and acquire a T1 map, cerebral blood flow, a PD map, a T2 star map, well, we have to improve the efficiency of our techniques because they can't be in there forever. So that's one of our main objectives. And there's already been a lot of work done in this in the field, but we need to push it further. Another objective for my lab, who's only like a year old, so that's why I'm not presenting too much new data here today, is to work on biophysical modeling. So really taking these multiple contrasts and fitting them together in a model to try to really map the microstructural features of interest. So I'll just show one example by the group of Marrakesh, which I quite like, and it's relevance to the data I showed today. What they do is that they acquire a R1, so 1 over T1 map, R2 star map, and a map of the magnetic susceptibility. Okay? And they know that all these features are related to the amount of iron and the amount of myelin that's in the tissue. So if we know in advance what the relaxivities are of myelin and of iron, we can then put that into linear equations and extract actual myelin and iron maps. So this was kind of a proof of concept. They haven't done extensive validation yet, but it's the type of direction that the field is going into and that my lab is very much interested in. A very different approach is to not use models at all and to do what I would call multi-dimensional imaging. So here, instead of acquiring a T1 map and like a diffusion image separately, for example, you really acquire those modalities together. So what we're looking at here, if you just look at one axis, for example, is really a spectrum of T1 times in a single voxel. So for the different spins that are there, they may have different T1 times in a single voxel depending on the environment that they're in, right? And then you do the same thing for another contrast. So here, let's say here, looking at the diffusivity, okay? To do this, you need to basically combine these contrast mechanisms while you're acquiring the data. So it's a much more extensive data acquisition, but it allows you to kind of understand the microenvironments that these spins are in without using models. So it's another direction that we're interested in going in. And then finally, a last objective that we'll be working on, that we started working on actually at the MNI, is animal imaging. So from an engineering standpoint, obviously we want to do animal imaging to validate all the new techniques that we're developing for human scanning. Because we basically scan either the mice or the non-human primates, and we can do extensive histology work afterwards to make sure that we're interpreting our data correctly. From the point of view of a neuroscientist, I'm actually using these animal experiments very differently, where I temporarily accept that our imaging techniques are not perfectly specific to myelin content, and I try to win back that specificity by using animal models. Animal models where we can start to play with the myelination, allow it, stop new oligodendrocytes from differentiating and forming new myelin sheaths, for example, and these types of things. And there we can then use MRI to image the animals longitudinally. So we regain our specificity by the animal models. We have MRI to look at longitudinal changes, which means that we can start to look at causal effects of myelin on behavior and cognition. So that's about it. Kind of gives you an idea of the directions that we're going into. And I'd like to take time to thank all of my collaborators, including Eve for the earlier work that was done at the MNI. And thank you for your attention.
Thank you for a very interesting seminar. Questions? Yana?
Thanks for a very engaging talk. I'm just curious, when you did the hypercapnia,
The increased CO2?
yeah,
Yep.
study,
Yep.
did you have trouble with the ethics board? How did you deal with that?
No. So, yes and no. As in, we absolutely have to have ethics, of course, and we only do it. So in this case, I think we went up to 5% CO2. So it's in a very safe range. I've done these myself. I've gone up to 10% myself. Not as comfortable. You can start to hyperventilate. Within at 5%, you're fine. It's really like you're going for a brisk walk. That's kind of the feeling that you get.
Yeah, it's a very vague question in a way.
Mm-hmm.
So when you start doing multimodality imaging, the time to take that image is longer. What about
Yes.
motion?
Okay. So we deal with motion in different ways. So sometimes when we do imaging, we'll acquire a T1 map, and then a T2 map, and then a PE map. So basically, the actual time that it takes to acquire one image isn't necessarily longer, it's just they're acquiring more of them. Some people will then just align the images after the fact, take their T1 image or T2 image, align them. Other people will run quick scans in between to realign the field of view to make sure that you're really acquiring these images in exactly the same way. There's some types of sequences, like, if you're looking at diffusion imaging, actually you're acquiring many images in a very fast way, an image for each diffusion encoding direction. And you can correct for motion between each of those images, the same way you do for functional MRI. So that's less of a problem. I'd say the real problem is when you're going for resolution, because then your scan time for a single image just gets longer. And there's a lot of work being done in that field using either cameras. So it's like cameras that are mounted in the bore or sensors. And there, what's nice is that it's kind of tied into the image acquisition. So what will happen is within every TR, I don't know if you're familiar with imaging sequences, you'll have a small dead time where you basically update the field of view real time of the images you're being acquired. So you can either use extrinsic information like the camera, or you can actually also acquire a very low resolution image during that time and do the image registration inside and update that way. So there's two different ways of getting the information to update the field of view. But
you don't see this really as an impediment? You don't see this really as an impediment,
I do.
The motion?
Yeah, I definitely do. Yeah. There's been really great work done by, in Boston, I'm going to say Tisdale, I forget who the senior author is on it, where they do perspective motion correction using the navigators. So this is the really low resolution images that are acquired throughout your scan to update the field of view. And they basically, what's nice is that they can acquire those navigators and they can choose to either update the field of view or not update the field of view. So effectively, they have an index of subject motion, right? And they basically did this in subjects and then looked at the impact that it could have on, group analyses of cortical thickness, gray matter volume, these types of things. And even in images where, we look at them afterwards and we're like, yeah, that's great, it passes my QA, there are subtle effects because it can just be a little blurring and that will affect your morphometry down the line. So I think that in cases where you're looking at depression, schizophrenia, honestly, so many diseases where the motion, like ASD, for example, autism, well, some kids may be more agitated, ADHD, the kids may be more agitated. Patients who all of a sudden take, what's the word I'm looking for? Medication that makes them more calm. Well, then you may have an impact, if you scan them before and after, but it may simply be that they stop moving in the scanner. So I do think it's a really big issue, but we're trying to address it. Yeah.
So for MS, you can use this for diagnosis, but is it also used for prediction of the outcomes of the patient, like how quickly they are degenerate or things like that? Or?
Yeah. So in MS, MRI is used extensively. It's part of the diagnostic criteria and has been for many years. At this point, I think that the research community is really trying to use it to monitor disease progression and to monitor the impact of therapies. So, I think that so I'm not a neurologist, but my understanding is that basically there's a lot of drugs out there that will help you in the first part of the disease where you have a relapsing, remitting course, right?